Hello. Hello.
उसे घर की दहलीज लांघते वक्त एक सेकंड
So very good afternoon. So people are joining this window. So those who are not getting this slot, then they can join in the YouTube live stream. That this link also be shared both in the mail and also in the group, WhatsApp group. So we'll start in a few minutes. So I request the participants, please mute yourself. Yes, ma'am. So after the 100 participants who will joining will be redirected into this YouTube link. So the participant can view the search whatever you have. Dear participants, sorry for the some technical issue because of the editing delay. Since we're depending on that internet, so it's getting some interruption. We'll be beginning in a few minutes. So those are waiting in the waiting room in the Zoom. They asked to join in the YouTube link, please. Okay, very good afternoon to all of you. So am I audible? Any response, please? Yeah. Okay. So we begin with this International eLecture workshop on innovation in biotechnology. So with the grace of God Almighty, we like to inaugurate the International eLecture workshop on innovation in biotechnology on today, January 19 and 20 of this year. So this is organized by the School of Life Sciences, with Rahman, Crescent Institute of Science and Technology, Chennai, and Crescent Educational Institutions, Madurai campus, Tamil Nadu. That is association with the Biotech Research Society, India, that is BRSI. So I take this opportunity to welcome the chief guest. Before that, I welcome the management persons and then Vice Chancellor of our university, Registrar, and our Dean School of Life Sciences and convener of this event. And also I welcome the chief guest of today, Dr. Rahman Sharif. He is a project leader at the European Bioinformatics Institute, European Molecular Biology Laboratory, that is EMBL, Cambridge, UK. And Dr. Saravana Kumar Kandasamy, who is a research professor at Department of Biohealth Convergence, Kangwon National University, Republic of Korea. The next today, Dr. Paul Loksho, one of the present his research is from the University of Nottingham, Malaysian campus. So today we're gonna have two talks. After that, I just briefing out this event before all join together. So today we're gonna have two talks from Rahman Sharif followed by Dr. Sarwan Kumar, and after that we're gonna have feedback link will be shared and also be shared in the Zoom and also be in the YouTube. So you can find this program schedule of today and tomorrow. So with this, I'd like to So I request our Dean School of Life Sciences and convener of the event to present the presidential address to this gathering.
Good afternoon to all of you. Can you hear me now? Now?
Yes, ma'am.
Okay. I think because of the internet, it's really, there is a problem here. I'm really sorry for that. Can I start? Can you hear me?
Yes, ma'am.
Okay. Good afternoon to all of you. The biggest innovations of the 21st century will be at the intersection of biology and technology. A new era is just beginning. This quote is by Steve Jobs. With this quote, I welcome you all on behalf of School of Life Sciences, BS Abdur Rahman, Crescent Institute of Science and Technology, for this international eLecture workshop on innovation in biotechnology, organized in association. You all know that biotechnology is a vast area of research from the basic to the applied level that includes study of numerous branches like molecular biology, recombinant DNA technology, microbial biotechnology, agricultural biotechnology, then immunotechnology, environmental technology, bioinformatics, proteomics, and genomics. Biotechnology basically aims to improve the quality of products for the sake of human well-being. The main target of biotechnology is to exploit the genetic material of microorganisms and multicellular organisms like plants, human beings, animals to obtain the desired product for the betterment of human life. The concept of biotechnology evolved with the study of microorganisms and their applications in many areas such as fermentation, medicine, agriculture, industry, and environment. Later on, many researchers were carried out various kind of experiments, and that led to the invention of transgenic plants, microorganisms, and recent scenario shows the vigorous growth in this scientific field aimed towards the development of more effective preventive measures diseases by genome sequencing technology. The development of insulin, the growth hormone, molecular modification of organisms such as hepatitis B or some of the mainstays of biotechnology, and its alliance with the genetic engineering. The revolution of new smart materials hands in hand with biotechnology has only just begun. It plants that can change the color when they detect an explosive or any pollution, clothing and footwear made with synthetic spider web, etc. Through bioremediation process, very useful for ecological recovery. The catabolic properties of microbes, fungi, plants, and enzymes are used to restore contaminated ecosystems. Biotechnological innovations are already part of our daily lives, and we find them in pharmaceuticals, pharmacies, food markets, among many others. Spearheads in the fight against the COVID-19, which is a global pandemic, since it helps to decode the virus genome and understand how our body's defense mechanisms work against infectious agents. This also led to the discovery of vaccines and also to control this COVID-19 spread. Biotechnology will therefore play a crucial role in the society of the future in preventing. This is just one of its many applications. And this lecture workshop will be focusing on innovations in biotechnology and its applications, and is aimed to provide up-to-date findings and covering different research areas in biotechnology, including lectures by immunoscientists. Dr. Rahman, project leader from European Bioinformatics Institute, European Molecular Biology Laboratory, Cambridgeshire, UK. Dr. Saravana Kumar, research professor from Department of Biohealth Convergence, Kangwon National University, Republic of Korea. Dr. Paul Loksho, director from Sustainable Food Processing Center and Future Foods Malaysia, University of Nottingham, Malaysia campus, have accepted to deliver special lectures in the busy schedule. I thank all the invited speakers for sharing their knowledge and time. We received overwhelming response from India as well as from other countries. We thank all the participants for joining hands with us to share their knowledge. I hope this workshop will help in the enrichment of interdisciplinary knowledge and a global vision to the research work. Stay safe and stay healthy. Thank you.
Yes, ma'am. Yes. Thank you so much. Yes.
Right. Good afternoon, everyone. Can everyone hear me well? I guess I don't have any acoustic problems. If you don't hear me well, or if you don't see my screen, can you raise your hand? Okay, so no one is raising their hand. So I think you could all hear me well and see my screen. Okay. Maybe an answer at this stage will be helpful. Can anyone confirm that you could hear me? Okay, there's a chat. Let me check the chat. Yes, okay, perfect. Thank you. Thank you, guys. I think I got 3 yes. And I hope I'm not having any acoustic issues. Okay, let's begin. All right, thanks to the organizers for inviting me. And apologies for the delay. I think there was some technical issue from the organizer's side that you did hear well. I hope that's not on my side. Right, so as introduced, I'm Sharif. I'm a project leader at the European Bioinformatics Institute, which is part of the European Molecular Biology Laboratory in Cambridge, in UK. So in today's talk, we'll discuss about the dynamics of cell signaling network and the lessons we can learn by imaging them. Right. So as many of you know, a living cell is a dynamic system. It's very smart, it's very clever. It can understand the environment and respond accordingly. So a cell in the eye will help you to achieve your vision properly, and a cell in the digestive system will secrete digestive enzymes and digest your food and help in the digestion, unless a neuronal cell will help in the transmission of signals. So every cell is specialized to do a certain specific function, and they are very clever in the way that they can respond according to the need of that tissue. They do this through the information processing unit, which is the cell signaling network. The network where different proteins are connected and talk to each other, and they collectively work together to decide what the cells should do, and they help the cells in understanding the extracellular stimuli and use the stimuli, process the stimulus signal, and then help the cell respond to the stimulus, execute cellular functions. So these actions or functions can include a gene expression, it can be secretion of protein, or it can be changes in the cytoskeleton in order to change the morphology of the cell, it can be growth of the cell, proliferation of the cell, and so on. So this cell signaling circuit or the protein networks control how cell respond or how cell behave. Right. So it is very important for scientists to understand this so that you can engineer it better and you can help solve diseases, because most diseases arise when the signaling circuit fail to do its proper or intended function. These cell signaling circuits are also very crucial because they connect the input signal with the output, which is the response of the cell, and it also helps in tuning the response. So if a cell is given certain input signal or growth factor, it needs to decide how many times it has to divide and proliferate, or how much protein it should segregate. So there is this link in quantitative aspect, and this is also determined by this cell signaling network, and also the dynamics. So how long the cell should secrete a protein given a signal. So this is also determined by the cell signaling circuit. Of course, the morphology and the proliferation of the cell, as we discussed. So there are several cell signaling networks. And of course, as I mentioned, cell signaling circuits are nothing but protein networks. The different proteins, they're talking to each other to execute certain function. So in this talk, I'm going to focus on number one, cell matrix addition sites, and the protein networks in the cell matrix addition sites. So what are cell matrix addition sites? These are macromolecular structures that helps attachment of the cell to the extracellular matrix. So these structures are very large along the plasma membrane. They anchor the cells to the extracellular matrix. They can sense the rigidity or the strength of the extracellular matrix and the signals around the cell. and they can modulate the extracellular matrix itself. I mean, they perform various functions besides attachment of the cell to the extracellular matrix. They help in migration, which is one of the important processes during embryonic development. And it's also important for wound healing. And when this migration is also critical in cancer where there's metastasis. So this cell matrix adhesion sites play an important role in various biological processes, such as controlling morphologies. It can also sense environmental signals, as I mentioned earlier, and by that means it can control the fate of the cell. Interestingly, these cell matrix adhesion sites are made up of more than 150 different proteins. So it's not the same protein, it's 150 different types of proteins. And these different proteins can be put together in different configuration to form different types of adhesion sites. That's why they are very heterogeneous in structures and in both in structure and function. And also these adhesion sites are highly dynamic, which means they are rapidly formed in the cell and rapidly disassemble in the cell. So when the cell is moving, you can see adhesion sites formed in the front edge and then disassemble in the rear edge. So it's very interesting to study these protein networks and how they function, how they come together to form different molecular structures, and how these structures form different cellular functions. So I want to explore the structure and function relationship of protein networks in adhesion sites. Okay, so one of the hypotheses or assumption is that the adhesion sites are heterogeneous. So it means that the cell has diverse condition within the different regions of the cell. So due to the different condition, different protein networks are formed and they perform different function. So in the front side of the cell, you can have different condition which leads to composition or protein network which performs function that is anchoring and propelling to move forward, used to propel and move forward. And the rear edge, there are adhesion sites which are of different network structure because the conditions that are different, that favors different network structure, and they are help in detachment and pulling the cells from the rear edge. So the idea is to follow these migrating cells, understand the protein networks, how they are formed and their function, and their role in migration of the cell. Right. So the main objective, to repeat again, we have cells in which we have cytoplasm where all proteins are present in this pool, and cell is picking up these proteins and building different structures to perform different functions. So this is just an analogy, but cell is actually building different types of adhesion sites. We're picking different combinations of proteins together, and they use those adhesion sites to perform different functions. So we try to explore the structure of the adhesion sites and thereby then the function of these different structures. Okay, so we want to image these proteins. Of course, that's the only way we can see where these proteins are localized and how they come together and function. So we need a microscopy-based approach where we are looking at the proteins of our interest. So in this work, to understand the protein and follow the protein, we cannot just image the nascent protein or the endogenous protein. We need to ectopically express the protein with a fluorescent tag. So there are fluorescent proteins, engineered proteins, which provide a signal that can be measured under the microscope. Just as you know, there are green fluorescent proteins which are often used to find the localization of a protein. So we can take one of these fluorescent proteins and tag them with our protein. So the way we do is that you take a vector, clone the gene or protein of your interest, and then following that you clone the fluorescent protein, this gene. So when you express this vector, you have a hybrid protein which expresses your protein of interest along with this fluorescent protein tagged to it at the same time. So by this way, wherever your protein of interest moves in the cell or localizes, the fluorescent protein also moves and localizes, then you can visualize the localization of your protein of interest under the microscope. For example, if you have tubulin, you can see the tubulin binding to the microtubules, and you have this nice structure visualized. And if you have actin, you can visualize actin tagged to fluorophore, and you can also use this to visualize various organelles and structures. Right. So in our case, we want to image many proteins, and we have to choose which protein we want to image to study the protein networks. Of course, we cannot image all the proteins. It's going to be really hard because spectrally there are not many fluorescent proteins that can be imaged simultaneously. So we have to make a choice. So then we decided to look at 4 proteins, which is zixin, vinculin, focalization kinase, FAC, and paxillin. So zixin is involved in the actin regulatory layer. So of the adhesion sites are attached to the actin filaments, and dixin regulates those. And then comes the force transductor. This is the layer of inside that senses force in the extracellular matrix and the addition and the actin. So can know if the stiff or soft, and so vinculin is there. So we chose vinculin, and then we look at the interin signaling layer. The interin of the protein helps the attachment of that to the extracellular matrix. So we have two key players, focal addition kinase and addition sites, and we machines and subject them to actomyosin inhibition for the inhibition of force, and this integrate actomyosin. This this inhibition sites because they need force to be intact. When the actomyosin when the actin fibers are lost, actomyosin contraction force is lost, the addition site is assembled. So we can use this inhibitor to inhibit the force and let the addition site disassemble, and then wash the drug, and then the addition site assemble again. We can do this process twice so that we can understand the nature of assembly and disassembly of these addition sites and understand their function, how they perform various cellular functions through the assembly and disassembly. Right. So now we want to image these 4 proteins. So to do this, I tagged these 4 proteins with 4 different fluorescent proteins of interest. We chose MK2, citrine, TFP, and MTAG BFP. They have different fluorescent color. They are excited different regions. So we have 4 different hybrid proteins. Of course, the fluorescence wasn't bright enough because we are measuring a very narrow region in the spectrum. So we have to sometimes use a tandem fluorophore, which means you have 2 fluorescent proteins stacked to 1 protein of your interest. For example, here with single zixin, we have 2 MK2, so that we have bright signal to measure. So through this approach, we can measure 4 different proteins, and these 4 proteins are localized in adhesion sites in a very small structures. So this is incredibly difficult, but also at the end very useful, because 4 proteins can image simultaneously together. And this was a very early study, or the first study to image 4 proteins simultaneously in the very same structure with high spatial and temporal resolution. Okay, so we got images of these proteins. We can image them, how the proteins are localized in the adhesion sites. Now, we want to quantify them. Of course, they look all very similar, but the levels are different in the adhesion sites. They are different in the front and the rear of the edge of the adhesion site, or the front and rear edge of the cell itself. So to understand this better, we have to quantify these images, the microscopic images, right? So we have this 4 color lifestyle images with high spatial and temporal resolution. Now, to process them, we developed a set of tools that can remove noise, remove background from these images, so that we can quantify them and then measure the level of different proteins at different parts of the cell and do a quantitative analysis, right? So we have several tools. Number 1, correction for chromatic aberrations. Sometimes light do not pass through the same direction. They kind of a little bit shift or distorted by the lens. So we have to correct for that, so that all the 4 images are aligned with each other. And then we have to do background correction, because there is some noise in the background. We have to remove them. And then there's also bleaching, because when you shine laser while imaging, you also bleach the cell. You have to fix that bleaching. And you have to do high and low pass filtration to remove noise, high frequency and low frequency noises in the cell. Finally, you have to do a lateral shift correction, because it's a stage in the microscope that can get shifted. So you need to help the microscope or the data to be clean. So you need to shift the images if this by mistake the stage in the microscope moves, right? So these are all automated, but then you also have we also build a graphical user interface to do this shifting and imaging. Right, coming back to our main question. So now we measure 4 proteins. Of course, we have many proteins, but we can only measure 4. Now we want to know how these 4 proteins are used by the cell to build adhesion sites of different structures and what kind of functions they perform, right? So, right, let's look at that. So to do this, to understand this, we're going to use a computational approach or a quantitative approach, which is called compositional imaging. This approach helps us to visualize protein networks and their states and the dynamics. Right, so I'm going to explain this approach. Instead of 4 proteins, I'm going to show this example in 3 proteins, so it's easy for you to follow. So let's say I'm imaging 3 proteins here, Paxil, Zixin, and Wasp in three different colors. And of course, we can look at them individually, or we can look at them superimposed. We can take a time course of these images so that we know how addition size change. But we will not have any meaningful information what are the protein networks. So to know or understand the protein networks, what I'm going to do is take every pixel from these images and build or understand what are the different composition exist. So every image is made up of pixels. So if you zoom the image to the level you can, as much as you can, then you will end up with several pixels. Or it is like let's say an Excel sheet where you have values for each cell. So, and then this is going to collectively tell the level of protein in that particular region. So every region in this image will have a number that tells what is the level of that protein that is shown as an image to us. So basically, from every pixel in these images, we will know the level, each of the image, we will know the level of the corresponding protein. So we will get the level of Paxil from this first set of images, and the level of Zixin in the second, and the Wasp from the third. From this particular region, this dot, this one dot, yeah. So then we can place this in a 3-dimensional space called compositional space, where the dimension corresponds to the level of these 3 proteins. So we can collectively take all the pixels from these images where we have addition sites, and then put them in this compositional space. So then we will end up with a cloud of various pixels. We can group these pixels and find those which are similar, which means we will cluster them and try to find what is the characteristic composition of these clusters. So I perform hierarchical clustering to find the 4 clusters A, B, C, D. I just label them, and then I can ask what is the mean composition or the mean or average level of the 4 proteins in these structures. So I can measure that. I can get the compositional signatures for these clusters, and then I can now spatially map them back, because I know where these pixels are coming from. I can spatially map them back in the images and understand how they are spatially organized and how they change over time. So here's an image. Now we have superimposed image, which is now converted into a compositional image where we have different structures which are corresponding to different compositions or protein networks. So this will help us understand different protein networks in the cell, the spatial organization, and how they change over time. So let's show you the schematic. So what we did, we took every single region in the image and took the region of all 3 proteins, and we put them in this 3-dimensional space where the dimension correspond to the level of each protein, and try to find clusters which are having similar properties. And we average out the mean intensity to find the signatures, and then we map them back in the cell to find out what is the spatial organization of these clusters and how they change dynamically. All right. So in the actual experiment, we can measure different proteins. For example, here in this example, I measured Zixin alpha-actinin. It's also part of the actin filament. And vinculin, Paxilin, I could identify 8 different clusters involved in these addition sites and these filaments. We can map them back to the cell. As you can see, these structures are nicely organized. So each composition here is a protein network, right? So each of them is a protein network, and then we know how they are spatially organized in the cell. And what we can see, as a movie, we can nicely visualize the dynamics of these protein networks through this compositional imaging, and we can understand what are the different composition of protein networks, how they are spatially distributed, and how they change over time, what kind of structures are present in the front edge of the cell or the rear edge of the cell, and so on and so forth. So we can now look even more closer into single addition sites and try to understand these in a better way. For example, the addition sites have a very nice or clear pattern of organization of protein network. So in the front tip of the addition site, which is towards the cell center, you always have a protein network that is high in alpha-actinin, and then the edge, which is towards the cell periphery, has a protein network that is rich in Paxilin. So there is a kind of internal organization of protein networks within the addition sites itself. And we can also look at this migrating cell and then see the organizational dynamics of the protein networks in the trailing edge and the leading edge of these cells. Right, so coming back to the main proteins of interest and topic, now we can subject these cells which are expressing all these 4 proteins and repeat the treatment of lock inhibition and see maybe this induces assembly of the addition site, assembly of the addition sites a couple of times, and we can monitor the changes and find the compositional signatures of the network protein networks and map them spatially. And we could identify different protein networks. I will not go further details here. Maybe okay. So you can see that initially addition sites are rich in zixin, but then when you inhibit them with when you treat them with the lock inhibitor and they disassemble, the new addition sites are again formed with the high zixin level. So you can see that, but then when in between, when you do not have addition sites or you have focal addition complexes, they do not have the zixin-rich structures. Right. So I mean, I'm not going to go into further details here due to time constraint, but you can read this manuscript to know more. Right. So we want to understand the structure function relationship. Like so far, what we have done is try to understand what are the different types of protein networks. Right. So for example, if you look at this one here, where Mitosis, I can tell that this one is made up of 2 Legos of brown color and 3 Legos of yellow color. So there's this 2:3 ratio between these two proteins, and that's what they form the structure of one. So that's what we have done. So I know that these addition sites, we identify different protein networks, and we know what are the proteins in that protein network and what's the ratio between them. We do not know how they are formed. So to know how they are formed, we need to see how different proteins enter into the addition sites, right? So to do that, I tracked individual addition sites, which means you have a computational tool that's going to track, find each and every object in the microscopic image, follow it over time, and then see how different proteins enter and leave the addition sites, so you know what's happening to them. And this can be quantified before and after perturbation, during the disassembly and assembly of the addition sites, and use this information to understand how addition sites are built and how they are disassembled. So this is all helpful possible because of the multicolor live cell imaging that we perform. So thanks to imaging and the quantitative analysis. So in this manuscript, we concluded, I'm not going into details here again. So we concluded how addition sites can sense multiple cues such as mechanical tension or signaling or matrix composition, and then use it to do a modular assembly of the addition site and perform multiple tasking such as addition strength control, actin modulation, force transduction, signaling, induction, and matrix modulation. So if you go into the manuscript, you can get more details, but for now, I would like to end the discussion here on this particular topic. But to move on further, I have another example of addition sites, which is the role of protein networks in embryonic development, how we can use imaging to understand these. Right. So many of you know that embryonic stem cells are pluripotent stem cells. They have potency to develop into various germ layers, and it's incredibly important to understand them for various engineering applications, such as regenerative medicine. So we can, to understand the changes in the embryonic stem cells, we can grow them in the lab. So there are stem cell lines such as H1. We can grow them with different growth factors, and we can induce differentiation of these cells into various cell lines. For example, if you differentiate the cells with the BMP4 growth factor, then you can get cardiomyocytes or trophoblast-like cells. What we could do is we can follow every single cell through these images and understand how ES cells or embryonic stem cells become somatic cells, and we can try to infer the networks that control the embryonic cells and the differentiator cells and how they change during the differentiation process. Right. So to do this, first we need to track these cells. So I've developed an algorithm which is going to identify every single cell and track them. And whenever a cell divides, we can follow the cells dividing into two daughter cells, and we can track what's happening in those two daughter cells as well. You can see sometimes the cells divide into two, and we can quantify the level of proteins in these cells, how they enter and leave leave the cells, and then try to infer the protein networks again, how they execute different functions in these cells, from differentiation to cell division and cell fate determination. For example, what you see here is CDT1, a protein that is increasing during the cell cycle. When the CDT1 increases the peak, the cells start to enter from G1 to S phase. So we follow the cells, track their family or the daughter cells, and we can understand cellular decisions. So sometimes when you have two daughter cells, they can both divide at the same time, and the protein networks are maybe synchronized. In some cases, this cannot be the same decision. In some cases, one cell divides first, another cell divides a bit later, or in some cases, that is asymmetric division. One of the two daughter cells divide and other daughter cells do not divide. So we can use this quantitative imaging to follow these single cells, quantitative imaging analysis, follow these single cells and use them to understand their protein networks and the behavior better. Okay, so there are multiple approaches we can use to do this. We can either fix the cells, stain them with different antibodies and image them, or follow the live cells, expressing topically various protein of interest or sensors and image them, and segment them using quantitative image analysis and do several analysis to understand the system better. I'm going to give you one example here. So we can ask, when does embryonic stem cell become somatic cell, by following every single cell and looking at the level of the preponderance markers such as OCT4, and we know when exactly at single cell level these fluorescent markers are lost. So as you see, also we can understand the dynamics of the pattern. So initially, embryonic stem cells are in colonies; they are very high in OCT4 level, but upon differentiation, after adding BMP4, they slowly start to lose these OCT4 levels, and we can look at every single cells how they start losing OCT4, and you can also see that there's a clear pattern. Cells start losing the OCT4 from the periphery, and then this loss or differentiation then moves toward the center, and every cells in the colony differentiate. So we can make single cell quantification, understand dynamics and the signaling networks better in these cases to do quantitative imaging and image analysis. So I'm going to show you another example here. So we can also look at structures, not just, we can also look at the proteins which are important for various activities. So for example, here we can look at cell cycle regulators and use them, image cell cycle, CDK, image sensors which can report the activity of the proteins such as CDK1, which is important, or a key cell cycle regulator. We can follow every single cell and quantify the level of these sensor. So this is a sensor which we use here, which is at the low level of CDK1 activity. This sensor protein is present in the cytoplasm, and the cells get activated. These proteins, this sensor protein moves into the nucleus. So by quantifying the cells, imaging the cells and quantifying the nuclear intensity of the sensor, we can measure the activity of CDK1 in cells. So as you see here, there is a peak. It means that the cells entered into high CDK1 activity, and then this decreased. The high CDK1 activity means the cells enter into mitosis, and then the fall means the cells then divide and become a daughter cell. So we can follow cell cycle master regulators activity in every single cell or any other protein's activity and understand them quantitatively. So we can also express fluorescent biosensors that can help us to understand the cell cycle fate, in which phase state the cell cycle is, either it is in G1, S, G2, or M, and we can then try to understand the protein networks in these cell states and how they help the cell to move from one state to the another. And this is crucial because cell cycle is something that is dysregulated in cancer. Understanding the networks and their dysregulation during this cell cycle, different cell cycle phases, will help us to potentially identify new treatment or targets in the network to tackle growth of cancer cells. Okay, not going to go into further details. So once we have these sort of quantitative imaging from single cells, of course, we can analyze them quantitatively, these networks quantitatively, and infer what's happening. But we can also understand the mechanism through modeling, right? I will give you an example here, which we used to, where we used to understand the mitosis using the quantitative imaging and mathematical modeling. So just to give you a flavor how this kind of approaches can go hand in hand and help us understanding the biological system better. So one thing, as you want to, talk to you about is the mitosis. So cell cycle, as you know, all has 4 different phases: G1, S, G2, M. Of course, some cells have very long cell cycle. Some cells have very short cell cycle. For example, ES cells, as I mentioned earlier, they have a short cell cycle, and the differentiated cells will have very long cell cycles, and on average, maybe 20 to 24 hours every cell divides. But in this entire, although there is a difference in the cell cycle timing, the duration of mitosis is always pretty much constant in all the cells. In many, almost all the cell types as well. So no matter the cell takes very long to divide or very short to divide, the time cell spends on mitosis is always constant and very precise. So we wanted to know why the cells always spend very precise time in mitosis. Of course, if you look from outside, mitosis is an important process. A cell has to divide into 2 daughter cells and distribute everything it has got into 2 cells, inherit them properly so that everything is properly done. If something goes wrong during these partition, it can lead to cancer. But although it's a very crucial process, cell does it precisely, very nicely within a short interval, and it's almost like 40 minutes plus or minus 10 minutes. So why is it so precise? So we try to understand this. We of course image single cells, and we get the quantitative information from single cells. Now, to understand the protein networks better, we try to look at the network signaling network that controls the cell cycle and try to build a mathematical model. A mathematical model is a mathematical representation of the biological system using mathematical equations, and we can use this to run simulations to understand the dynamics, and then we can fit with the data and improve the model if needed. Once we fit the model and got a right model, we can make new prediction or make new in silico experiments to understand the biological system better. All right. So in this model, we perform several single cell simulation, and we perturb different network. We try to inhibit a protein. For example, we try to inhibit MAT2, which is important for the mitotic regulation in the spindle formation during the cell division, where the cells, 2 daughter cells, separate the daughter chromosomes. But when we inhibit the MAT2, still the mitosis is always constant. Cells still divide irrespective of that, and the mitosis time is pretty much similar or very short and precise. And of course, we tried several perturbation through this mathematical model, perform in silico experiments. And then we found out that if we perturb positive feedback in the network, which is through the V1, we can perturb the duration of the mitosis. We then performed experiment. And then in the experiment, we showed that when we use an shRNA for V1, then the mitosis duration is perturbed. So first we built a model, and we use the data from the single cell imaging, and we showed that mitosis is constant. And then we did perturbation in the more computational model, various experiments, and these experiments hinted that only the positive feedback loop can keep the mitosis modular and make it short and constant. And then we did experiment in the lab. This again proved the hypothesis from the mathematical model, and we showed that indeed the positive feedback in the small signaling network is what keeps the mitosis modular, short, as well as constant. Okay, to know more, you can read this manuscript and get further information. Right, so if you want to use the model itself, then you can go to Biomodels database, which is a database of mathematical models. You can get these models readily. These are the models of various protein networks, how they are connected with the mechanistic information, and you can quickly, these are curated models, so you can quickly run them in any of the modeling softwares, and many modeling software that support SBML, which is a standard, and understand the biological system better. Of course, not all these models were not based on imaging, but there are models which are based on imaging. There are models which are based on the other experimental data. So with this, I would like to end the discussion and acknowledge everyone involved. And thank you for listening, and I'm happy to take any questions if there are any.
Thank you so much, Dr. Sharif, for your great presentation. Very sorry for that, for the internet interruption. From that, you have done very nice presentation over there. Thank you. So, I'd like to welcome in any discussion. You can raise your hand, then I can allow you to unmute. Or you can type your question in the chat box as well. So participant, now you are allowed to ask any question. Now it's open for discussion.
Okay. I think people are a bit shy, or other questions. Maybe you can. Feel free to write to me if you have any questions here, and maybe I can also advise here. Say, I'm not doing lots of imaging these days, which I presented. So these are things which I did in the past, but I'm more now interested in modeling. I'm a bit of now mathematical modeler. So if any of you are interested in modeling, mathematical modeling, so you need to have sound background in mathematics, coding, and of course biology as well. So then you're welcome to apply for an internship or visit in our team. And of course, it's, we have paid internship. Of course, it's competitive, so you have to pass through an interview, which is where, and then called for this position. So so you should have a proper background. So if you are selected, you will be funded. It can be 3 months to 6 months internship, of course, but that's more specifically on mathematical modeling of biological systems.
Okay. Dr. Sharif, there is some question from Praveen. You can able to see that, or I can announce this?
Yeah, no, I can see that. So what are the free softwares available for quantitative imaging? Yeah, good question. So I will also type the answer here. So there are two which I would definitely recommend. Number one is Image. Oh, sorry. I have to send to everyone. Image. And then there's this tool called Cell Profiler. So these are two different tools which are quite commonly used, and you can also use MATLAB, but this requires a bit of advanced programming knowledge. Imaging is very common. Of course, if you know Image, Cell Profiler, if you are a PhD student, and if you're looking for PhD abroad or a postdoc position abroad, this could really help in getting a good position if you know any of these tools, and if you're applying to a cell biology lab. And there are several tutorials where you can learn how to use these softwares. Okay, so there was this next next question from Akansha Jain. So her question is, what's the scope of extending mathematical modeling to biofilms? Right, so of course you can apply mathematical models to various systems and processes. There's no limitation, of course. That applications or advantages general. The scope is broad, so you can pretty much express anything, any biological system, as long as you have data to build the model. I think you can use it to understand the efficiency of various microorganisms, and there is specifically a modeling approach called metabolic genome scale metabolic modeling that are used to build models of microorganisms. Maybe that can be potentially useful to study the biofilm. Maybe if you want to use that you can use that to engineer the organism or identify a drug against an organism. And there are several approaches or papers. For example, there was a recent paper to identify a drug for COVID using this genome scale metabolic model as well. You can pretty much apply this in various fields. It's commonly, mathematical modeling is also commonly applied in drug discovery in the pharmaceutical industry. For example, if you want to know the right dose of the drug and how often the drug should be given, those things are estimated using mathematical models. So the drug, whenever you are prescribed drug by the doctor will tell you take one in the morning, one in the evening, or twice a day, or once in a week. So these are all estimated using mathematical models. So the models are designed to define the rate at which the drug is metabolized in your body, the rate at which the drug is uptaken by the body, and these are called PK/PD modeling, pharmacokinetic, pharmacodynamic modeling, which are very commonly used in the pharmaceutical industry. So the application or the scope of mathematical modeling is quite broad. If you're interested, you can go to biomodels and just quickly search area of your interest, for example, biofilms or cancer. You will find several existing models, so you can use them and start building further from there.
Okay. Thank you. Sharif, I have one question. Okay. How come the cellular dynamics helps in tissue engineering?
How? Again, what was the question?
The cellular dynamics. Cell dynamics helps in tissue engineering. Okay,
so cell dynamics is it's important to understand the cell dynamics, right? So all we did is quantitative imaging, understand how the protein networks behave at different situations, and this is particularly important for tissue engineering, because when you have to put together several cells and artificially and try to build a tissue, or sometimes you need to know their behavior, number one, first, and then how they behave when they are in contact with other cells. I think understanding the cell signaling networks better would help to engineer them better. And of course, if you have mathematical models, you can also understand them better. For example, there are models, multicellular models. We are writing grant proposal on that. That we can do simulations. If you can guess or predict what will happen if you put different cells together. And of course, if you have imaging, this is going to help provide data, and then the model can help you understand the dynamics. And if the cells don't like each other, you can make these predictions already using mathematical model and use this information to improve the tissue engineering or a scaffold material that you're going to build. Yeah.
Okay, thank you, Sharif. You can maybe you can stop share, then we can proceed with other things. So I can share. Yeah. Because due to this technical issue, I could not introduce yourself here. Maybe I at least at first presentation I start give you about your credentials. Okay, actually, Dr. Rahman Sharif is obtained his PhD in Max Planck's Institute of Molecular Physiology from the Department of Systemic Cell Biology from Germany. He worked on spatial and temporal dynamics of protein networks in cell matrix adhesion sites. And he is a visiting scientist in Imperial College London Medical Research Council, and also from London, and also he got award from the CSC Local Hero Award of many years, but actually mentioned one in 2015 for organization postdoctoral programs. And his publication cumulatively 35 research articles in high reputed journals, and he has got many grants. At one of the time there is international grant for the myeloid landscape of NSC LC, it's worth of 600,000 euro. It's almost 5 crores. Yeah, 5 crores of project in Indian rupee. And he's interested, he's working on network remodeling drives human embryonic to somatic transition, lessons from quantitative imaging analysis tool development, embryonic stem cells rated the signal circuit to become the specialized cells or his specialized area that he is working now. So with this, I like to thank Rahman for his outstanding presentation. He says his certificate of his appreciation. Virtually, I'm presented now, then later I can send you. Thanks for coming and accepting our invitation to deliver this talk. Thank you, Sharif. I hope you accept this.
Thanks for thank you. Yeah. So just to mention that the grant was a joint grant, a collaborative grant. Yeah. Okay.
Yeah. That's what.
Okay. Thank you.
Thank you so much. Okay, we proceed to the next presentation and final of today's presentation is by Dr. Paramatma Kandasamy. He's going to talk about trichoderma from the ecology application in nanotechnology. So, brief about him. Actually, I have a very big CV, so I just made it into small. He obtained his PhD in marine biotechnology, where he worked on studies on mangroves derived fungus, that is especially trichoderma, and their biotechnological applications from Annam University. Later, he is a research professor from Kangwon National University, Chungcheong. Now, recently he got Brainful program where he is promoted as an innovator scientist, sponsored by NRF, that is National Research Foundation of Korea. And he won many awards. For the notable grant is the trichoderma metabolites loaded chitosan nanoparticle for cancer therapy. This is equivalent to 1.78 crores per project. And he have any other project as well in before he worked in China and here. And he is acting as an editorial board of many international journals and delivering talks internationally. And he filed 11 patents, 130 publications, and his cumulative impact factor is 442. And his gene bank accession is he deposited almost the fungal genes to the gene bank is 1,600 plus. So he has vast experience on fungal biology, and is a life member in the Mycological Society of India and many other society. And he also holding many specialists on fungus and also application in nanotechnology. So with these words, I'd like to call Saravanan to deliver his lecture. Saravanan, you can unmute and talk. Dr. Saravanan? Sorry. Yes, Dr. Saravanan, please, you can share your slides.
Yeah, thank you very much, Dr. Mubarak, for your nice introduction. Just a moment, I would like to share my slides.
A small announcement. After his talk, the today's feedback link will be shared both in YouTube link and also in this window. So participants have to fill after this talk. Thank you.
So can you hear me?
Yes, doctor. Yes.
Yeah. Okay. Thank you. Thank you very much for kind introduction about my candidature. So with your introduction, thank you very much for inviting me to give a talk on about trichoderma and also the little bit about the marine ecosystem and their associated of fungus, particularly trichoderma. So I directly, instead of wasting time, I directly go to the presentation. So actually this presentation deals with from the field to the immuno-nanotechnology, how the trichoderma is utilized in the different applications, and also some including immuno-nanotechnology, how it's emerging from the growth. So this is seems like I started this research from the 2009. So this is just a compilation of about 10 years research with some important insights and also important informations. How can we utilize the trichoderma, particularly fungus? So with this, first I give the basic introduction. Actually, the fungal strains can be isolated from the marine ecosystem as well as the terrestrial region. In marine ecosystem, fungal strains are involved in the variety of the functions it can utilize to be decomposing organic matters and also converting some kind of life cycle, food life chain in the marine ecosystem. For example, it may be co-regulating with the bacteria and also so many factors, and it can also allow to grow some wetland microbes as well as. So due to this, the fungal habitat will be supporting to the marine mangrove habitats and also wetland habitats. So we have done on the fungal from the marine ecosystem. Actually, in general, marine the fungus can play the important role, as I mentioned in the previous slide. It can be used in the nutrient cycling and the food webs. From the fungus can be isolated more than 1.5 million to over the 5 million, but likely we have isolated at present moment only the 10% when being stated from the including terrestrial and marine ecosystem. Particularly in the marine ecosystem, totally 7 phyla and 22 classes and 88 orders and 1,692 species have been identified. But this is just below 10% of the fungal identified so far in the marine ecosystem. In addition, you can able to see from the marine, the fungal strains being isolated from the various locations, particularly in the wetland ecosystem and also the estuarine and the coastal areas. You can able to see here the point out how many strains in the different locations being isolated from the Chinese coastal in the Yellow Sea region. They have been reported many fungal strains being isolated comparing to the other regions. It indicated that there was less fungal strains being studied in the other resources. So the other region also need in the indication to study more on the fungal strains, particularly in the coastal wetland ecosystem as well as in the marine ecosystem. So in addition, the fungal strains can be isolated in the various resources from the marine, including wetland ecosystem, for example, mangroves, sponges, algae. In some cases, it can be also identified as the surface as well as from the endopetically. So here you can see sediment, coral, sea animals, sea water, and one non-plants, because in the wetland ecosystem, we also have some marine plants. So we also included that as the non-mangrove plants. You can able to see the most of the studies were carried out. It's related to the mangrove ecosystem or sponges, either it is the endophytic or surface. In the algae also it's the endophytic or surface, and also on another case in the sediment. So it indicated the rest of the resources are not exclusively studied on the fungal isolation from the marine ecosystem. So this gives the new idea, new attention to identifying the fungal strains, since it have the various important applications. So come to our study. With this background, we have involved and interested to work on particularly trichoderma strains. Actually, this classical idea is just emerged when I'm working on the yeast, because in beginning I started research on the yeast. So when working on the yeast, I came to know trichoderma also being a good fungal group. Also, it can be used for the various applications. And the studies on the trichoderma is more common in the terrestrial region when comparing to the marine or wetland ecosystem. So I got an idea to work on this based on the literature. And we have tried to isolate the trichoderma from the various resources, as I mentioned in the previous, including all this. And here I give the more information on that. The marine species being reported on 1974, but since to date, the isolation and identification of marine trichoderma strains from the wetland ecosystem or marine ecosystem is too less. So it gives the more attention to work on more on trichoderma strains. So one more key feature is you can able to see here about 104 known trichoderma species has been reported in ISCatch, because this is kind of a database for trichoderma species. In this database, they have mentioned about 104 trichoderma species, including 20, in which only 21 was found from the marine ecosystem. It means mangrove or the another resources also be considered to isolate the marine fungus. Here you can able to see only these are the trichoderma strains being isolated and reported from the marine ecosystem, and these reports were confirmed that the mangrove habitat is the rich source for isolating the novel trichoderma species. So with this, we have started to isolating the trichoderma species from the various resources from the wetland ecosystem in Tamil Nadu. It's in southeast coast of India, in the Pichavaram forest. We have made a study on how to the ecological factors as well as the different locations, because as I mentioned in previous slides, the trichoderma strains have the prominent serving and also with food web chain in the mangrove or marine ecosystems. So we have tested from the two different locations. One is from the luxurious mangrove forest. Another one is the degrading mangrove forest. This means there was the two kind of this degrading mangrove forest means it's just started to growing the mangroves. So in these two locations we have compared in this study. So in this study, first we have isolated from the soil. In the soil, we have, as I mentioned, there is the two locations. We have found the more number of counts from the luxuriant mangrove site comparing to the degrading site. Also, we studied in the different depth. You can able to see here the counts of trichoderma were found more in until the 40 centimeter depth, but when comparing to the other depth where it is too less, or maybe no, or maybe one or two counts may be found. And in case of the seasons, you can able to see here the more were found from the monsoon season comparing to all the other two seasons. It's because of the water inlet, because the trichoderma strain has been used as the fertilizer. So the water inflow maybe have the vital role for getting the more colonies of trichoderma in monsoon. In addition, we also tested the trichoderma from the Vesturian water in the three different locations in order to confirm whether the trichoderma strains is a facultative form or it is the true marine fungus, because as it is a definition is facultative form means the fungal strains originally originated from the terrestrial region. It may be come to the marine Vesturian system or marine system and accumulating themselves to adapt the salt condition. In case of the true marine strain means the strain is originated in the marine environment. So in order to know how the inflow water influencing the marine strains, we can able to see here that trichoderma strains were found in the mangrove zones comparing to all the other zones. It is also indicated that to have the marine source is rich is also agreement with the previous research. And here you can able to see there is also some different time interval we have tested due to the photolytic activity. In the daytime, you can able to see very less fungal strains, but in the night hours, we can able to see the more fungal strains. And so this, in case of the season, we can also see the more in the monsoon season because of the water inflow. And in case of the trichoderma also, in case of decomposing leaves, we have isolated from the two kind of the mangrove species, and we isolated this experiment. What we done, we just collected the two kind of the mangrove species leaves, and we just artificially allow them to decompose in the marine environment. When decomposing, we have collected the liquids of the samples by every 8 days interval, and we have tested their colonies count of trichoderma, and we found the colony count were very less during the 0th day. And when the days of incubation is, when the days of decomposition is increased, the colony count were increased. This also we found more on the post monsoon comparing to, because this decomposition artificial experiment were carried out at 4 different seasons, and we found the more in the post monsoon. It's due to the inflow, because we just dig in the marine wetland ecosystem, and we pour some water and put some leaves inside that that well. So in that well, there is chance to adopt. There is chance to keep the water just coming from the other terrestrial environment. So that is the reason that maybe more counts from the post monsoon. And also we tested endophytically from the total of about all the mangrove species we found from the whichever mangrove ecosystem. In that we have tested, and we found the maximum fungal count were found in the Lumicera racemosa, and it have the first correlation with anthocyanin and flavonoids, because some pioneer studies shows that when trichoderma colonizing in the endophytically, it can trigger some innate immunity, and that kind of due to that reason there were the anthocyanin and flavonoid counts may be more. This the detail I will explain in future slides, upcoming slides. So with this, in the isolation, in case of the India, we have isolated a total of 256 trichoderma strains, in which about 12 species we have identified based on the ITS sequencing, and the sequencing were deposited in the NCBI. And followed by, we also studied in the same manner in China. We have collected the samples from the different provinces in China throughout the LOC, and we found we isolated a total of 252 strains. In here also we done the culture dependent and non-culture dependent two methods. One is culture non-dependent using the pyrosequencing. In this study, we found that when we pulling all data related to the traditional and pyrosequencing, we found a total of 252 isolates composed of 15 known and 2 unknown trichoderma species. And in this location, we found trichoderma atravide and trichoderma azanum found to be a maximum and rich in the study site. Among the study site, the Hangzhou from the Zhejiang province found to be more diversified and counts. On the other hand, we also studied from the Korea about the fungal trichoderma strains. We have here in terrestrial wetland ecosystem located in our near to our city, and another one from the coastal wetland system. It's located in another city, is just east coast of Korea. So in these two locations, we have collected the samples, soil and water samples, and we identified here. Actually, trichoderma strains have some difficulty in identification when we do the ITS gene sequencing. So what we done here, we just used the 2 gene sequencing, that is ITS and TEF's gene sequencing, and the gene sequencing may be the gene sequencing after obtaining the put together, and then we have to do the blasting analysis. Then we find the identical strains. Their identical strain and their corresponding reference strain were used to draw the phylogenetic tree. Based on this, we have find out a total of 19 in 2 different sampling sites in Korea. So, followed by in the ecological studies, when we correlating with all the, because each station we have also studied all the in situ in the laboratory meters, and we have done the correlation studies, and the correlation studies shows that the temperature and also the redox potential and the pH have the vital role in. So the drug are found to be higher in China comparing to the two and South Korea. And one more meaningful reason here, in China we have studied regular strains from the several sites. The number of sampling also is more than 1,000. But in case of India and South Korea, the number of samples is not that much. We have studied only limited, because since we are switching into the application studies, we have just established the resource and we focused on the advanced experiments. So that is also one of the reasons. So the comparison between the two different country or station, it's maybe depends upon number of the samples and also other conditions. So it needs the more exclusive studies when we want to come for the conclusion about which one country is the rich in record diversity. So in other case, all these gene sequencing, as I mentioned, we adapted to the DNA barcoding, and we have used the multiple gene sequencing to identify the Dragonderma strains. And then the gene sequencing were deposited in the NCBI gene bank. At present moment, we have deposited more than 1,650 strains of Dragonderma strains. It is means, here only not ITS alone. It is together. One is the ITS. About ITS gene sequencing, we have deposited about 1,650. And TF we deposited about 1,650 strains at present moment. So with this, we goes for the application. Actually, Dragonderma strains can be utilized in the several applications, including in the enzyme. For example, Dragonderma strains is being used as the cell factory to produce the several kind of cellulose enzyme. For example, cellulose enzyme, chitinase enzyme, or not. Most of the many of enzymes can be isolated from the Dragonderma strains, or using the Dragonderma strains be a cell factory to secrete some kind of enzymes. In addition, it can be used as the in bioenergy and agriculture as the biofertilizer, and back to metabolites in the two kind, one for in the agricultural research, and also in kind of biomedical research as the natural product being used for isolating some kind of novel metabolites to target some cancer as well as the bacterial infections. And also it being used for the bioremediation as well as the nanotechnological applications. So I summarized some interesting information on that. Here, firstly, when we got the total several kind of the Dragonderma strains, most of the strains we have subjected to the screening against some Fusarium stock rot disease. This is in anagonistic experiment we tested against the Fusarium graminearum. From the basic screening, we found one of the strain have the about 96.3% inhibition against the Fusarium graminearum, and therefore we chose that. And from that, we have isolated some secondary metabolites and tested the different concentration of metabolites against the Fusarium graminearum. We found that when the concentration is increased, the Fusarium graminearum growth were inhibited. So we confirmed this have the good ability to produce some metabolites to target the Fusarium graminearum. Followed by that same strain being tested in the in vivo plant experiments. In the plant experiment, we tested in the mice plant line, that we found that the Dragonderma treatment can increase the root and shoot length, also the root and shoot biomass, and reduce the disease. This also we have shown here when comparing to the 4 different treatment. For example, is untreated, and it was the Dragonderma and Fusarium graminearum alone, and the Dragonderma plus Fusarium graminearum treatment. And then we have found how much the Fusarium graminearum colonization is found when it is in the root. We have tested, and by using qPCR, we found that in the non-treated group there is only Fusarium graminearum were found, but in case of the treated group, Dragonderma as well as the Fusarium graminearum count may be disappeared. So this also we found, and the root colonization we have found using the GLP tagged gene, by tagging the GLP into the targeted strain, so that we can able to see how the Dragonderma strains can able to colonize in the root of mice, and how it is forming the interactively initiate the innate immunity of the plants. And followed by, when we give the treatment, how the other microbial other root microbiome, how it is having the interaction between the trichoderma. We have tested by using the biosequencing analysis. After treatment, the root soil samples were collected and tested their bacterial OTUs by using the biosequencing analysis. In that analysis, we found one of the bacteria, Acetobacteria, were found to be higher in the trichoderma treated group comparing to the non-treated group. It indicated that trichoderma strains can have the higher ability to trigger the Acetobacteria. Acetobacteria is phosphate solubilizing bacteria. This have the good ability when combined with the trichoderma. So this also we found the interesting information. Followed by, in the fungal OTUs, we can find more fungal OTU. We can able to see here a decreased count of Fusarium craminarium and comparing to the trichoderma count. So it indicated that when the trichoderma is treated to the soils of mice, there will be the trichoderma colonization were increased and their growth may be trigger their increased fold of trichoderma counts comparing to the Fusarium craminarium. So when we come with these all the questions, we may have the another question. So trichoderma is colonizing in mice, means how it is colonizing and what is the receptor, what is the eliciator, we need to find out. So for that we have made some experiment to find out how, because in one of the study we find the trichoderma eliciator like THPH1 and THPH2, it's kind of the cellulose gene. This maybe have the role to interacting with the mice roots. So we find out the
Dr. Saranam? Sorry for the interruption. He have some network issue, so he's joining now to share his slides. Participants, please wait. He's trying to join. And for tomorrow talk, the same time will be followed 2 p.m. You can join the same link, and the first 100% will be allowed. Then after that, will be redirected into the YouTube link. So as such, what happened today? But we hope next day we won't be have any network issues. And for the brief of today, tomorrow we have only a talk from the Minister Natufam from Malaysia. And for the, after that, we have inauguration of Association National, Nano and Biometric Association. So those who are interested in the joining in the association, you can show interest. Already we captured the information. Then we send you the a link to join for that. So Dr. Saraman has joined. He's sharing now. Yes, you can unmute and talk, Doctor.
Can you able to see my slides?
Yes, yes. Actually, you are left at 26th slide, where it is stopped in 26th slide.
Yeah, okay. Now, sorry for the inconvenience. Yes, sorry for the inconvenience. I think, I don't know, it shows some kind of unknown errors. Okay, anyway, let's go with the continuation. So actually, in as I mentioned here, is it okay? Can you able to hear me?
Yes, yes, you can. We are hearing you.
Because you know what I'm talking, the same sentence talk I'm getting again. I'm hearing again.
So parallelly, you open any YouTube window. If it is so, you can close that. Live streaming is going on. Is there anything?
Now is it okay?
Yes. We continue from the sixth. The slide is changing.
Slide is not changing.
Changing.
Some proof.
Go to the presentation mode.
Yeah.
Yeah, now bioethanol is there.
Yes. Yeah. So as I told in the previous slides, there is also some there we find that Trichoderma species can able to interact with the plants, and through the interaction, it can trigger the some kind of pathogen in some kind of plant immunity to target the pathogen interactions. So in this we can able to in the followed by we aimed to study the how the Trichoderma strains were interacting and how through it's interacting. So where we aim to study the receptor and eliciator. So based on that we used some of the experiments to find out what is the eliciator and receptor is responsible for interaction between Trichoderma and the host. From that study, we find there is the two genes from the Trichoderma have the interaction with the root. Similarly, from mice root there will be and also GLP3. These two genes are interacting with the Trichoderma species. Through this interaction, it may trigger the JAET upregulations in the leaves. Through this, the plant immunity were triggered and the pathogenicity of fungal strains will be controlled. And then the bioethanol production we have studied. In the bioethanol study, in the another application of Trichoderma strains, we used to isolate to study the bioethanol production. In this, you can able to see here the Trichoderma strains can able to produce some enzyme cellulase. By using the cellulase enzyme, we used to convert the sawdust into the sawdust is the multiple sugars and also kind of lignin and other component also there. So we have used some acid processing and using this enzyme to convert the simple sugars. After getting the simple sugars, we introduced the bioethanol production. In this way, we have utilized the Trichoderma strains for bioethanol productions, and followed by Trichoderma strains, we have in the bioethanol production studies, we have started in beginning with some terrestrial yeast strains. In that time, we have got about 12.8%. By using the different kind of methods, we have enhanced the bioethanol production, bioethanol yield, and when we using the dual fermentation, it is the combined fermentation with the Trichoderma as well as the yeast, we found about 64.3 gram per liter of bioethanol production. So it is the comparatively good and it gives a good production of the bioethanol production. And followed by the Trichoderma strains were used to for the bioremediation, for example, iron and also lead removal. In these two way, we have find bioethanol the biomass of Trichoderma being used as the absorbent to absorb some iron and lead from the aqueous solution. This is the biomass before the accumulation, and here you can see after the accumulation by using the EDS as well as the ICP, we find out the percentage of iron removal from the aqueous solution, and we found that Trichoderma treatment, the Trichoderma absorbent, when we used as the agent to remove the iron and the lead, we found about 88% of iron removal and 94.4% of lead removal. We also used the Trichoderma strains for removing the dye declaration. We found the these also have the function to degrade the dyes. And also we used to remove the Trichoderma strains to remove some blood stains. This also can able to see with the control and distilled water when we're using water to remove the stains. And detergent, when we comparing the Trichoderma derived enzymes which have the higher ability to remove the blood stains. And also Trichoderma strains being used to produce some chitin enzymes by fermenting the prawn waste as the source. When we fermenting together with chitin and with the chitin resource of the prawn and also fungal strains together, we got the crude chitinase enzyme. This also we purified, and the molecular weight we determined as the 49.49 kDa. Followed by we use the Trichoderma strains derived metabolites to treat some kind of the metabolites to use to test their antibacterial activity and also toxicity and cell viability. And the metabolite from the Trichoderma exhibited the excellent antibacterial activity against the Helicobacter pylori and Escherichia coli. Followed by this also inhibited the cancer cell line, the prostate cancer cell line. We found that it inhibit the prostate cancer cell line through the BCL2 and caspase-3 mechanism. And also followed by the another compound we isolated from the Trichoderma strains have the anti-skin cancer activity, we tested using that compound. We found that the compound can enter into the cell, and it causes some mitochondrial and also HSP19 protein expression, down regulating the protein expression and ROS production. Through this mechanism, it caused the apoptosis in the cells. So this mechanism we evaluated and published in the recent article. And then the trichoderma strains being used to synthesize some nanoparticles, and we used the AgNO3 and magnesium compound nanoparticle. In this nanoparticle, we tested their antibacterial and anti-cancer activity, and we shown here the anti-cancer activity against the lung cancer cell line. We found that that nanoparticle have the higher lung cancer activity. When you can able to see the treatment will inhibited the lung cancer proliferation. And then we also observed under the light microscopy and AOEB staining, you can able to see here the ROS production and the nucleus damage, as well as the higher necrosis cells were found in the AG and MGO-NPS treated group of the cells when comparing to the other untreated and treated with the magnesium nanoparticles. So followed by the trichoderma strains also used to synthesize the chitosan nanoparticle. The chitosan nanoparticle synthesized from the trichoderma strains also have the exhibited the good activity against the bacterial pathogens. This is the characterization we done by using the symptom analysis and UV as well as the EDS, followed by the activity shows that have the good activity to incubating. And followed by, we also synthesized the copper oxide nanoparticle. And the copper oxide nanoparticle we tested against to the lung cancer. This also induced through the capacity activation mechanism. And then we introduced the aptamer conjugated iron oxide nanoparticle. That iron oxide nanoparticle, where when aptamer is present is a kind of ligand can be developed to target the nucleon present on the cancer cell line. The nucleon only highly expressed in the cancer cell line when compared to the normal cell lines. So we have conjugating the aptamer on the iron oxide aptamer on the gold nanoparticle. Gold nanoparticle already synthesized using some fungal molecules. So when this protein in the protein coated, the fungal protein coated iron oxide nanoparticle conjugated to the aptamer, it have the promising activity against the cancer cell line through the apoptosis and mitochondrial membrane loss, as well as through targeting the some nucleon present in the cancer cell line. So this is also evidenced by the several experimental results. So come to the immunocancer therapy. Actually, the immunocancer therapy in general can work on the three kind of the mechanisms. One is a non-specific immune stimulation and T-cell transfer therapy and immune checkpoint inhibitors. The immuno compound, it means the immune system will be stimulated by using some compound. That compound being synthesized through some cells or organ present in the human system. And the compound, where whatever produced, that compound may stimulate the immune system to target the cancer cells. Through that mechanism, the immune stimulation mechanism also causes the cancer cell appletion. Followed by the T cell is kind of the great weapon to target the cancer cells. Actually, the T cell function may be controlled through the immune checkpoint inhibitors. Immune checkpoint. So immune checkpoint, when it is bind into the T cell in inactive, or it means switching off mode. So when it is switching off mode, the cancer cells may also can develop such a kind of immune checkpoints. That time the T cells unable to find or unable to differentiate the cancer and normal cells. So this reason it unable to target the cancer cell. So when we introducing the immune checkpoint inhibitors, that immune checkpoint inhibitors can stop the interaction between T cells and also the immune checkpoint in the cancer cell line. So through this mechanism, the T cells will activated and will target the cancer cells and cause the ablation. So this is the mechanism in basically occurred in the cancer immunotherapy. So followed by, for the instance, the cancer immunotherapy is originally originated in the 1980s, and it is developed in the by the William B. Coney in the 1918 to 1936. So this all kind of study is started. And then, for example, as I mentioned in the previous slide, is for example, the tumor cell. This is the T cell. For example, you can say from the tumor cell and the T cell, it have the PD1 and the PDL1. When it is having the interaction each other, that time it unable to kill the bacterial cell, because this also can have some checkpoint, immune checkpoint inhibitors. So immune checkpoint, that is a PDL1. So that when we introducing the immune checkpoint inhibitors like kind of antibody, anti-PDL1 or anti-PD1, that time it will block the interaction between PDL1 and the PD1. That time it will trigger the activity is cause the tumor cell death. So through this mechanism, it may be occurred. So in this context, we done a study on how we use the trichoderma derived nanoparticle to trigger the immunity. For example, in this study, we use the beta-glucan as the immunostimulant. Is commonly used in as the immunoadjuvant or immunostimulant to treat the cancer patient. So in that case, what we done in this study, we have developed the zinc oxide nanoparticle by using the trichoderma strains. Then we have conjugated with the beta-glucan. That beta-glucan will support to trigger their anti-cancer activity by interacting with the macrophages. So that we have characterized the nanoparticle by using scanning electron microscopy. It's found the spherical safe, and then we further tested with the TIM analysis also. Then we tested the size of the nanoparticle and the bare nanoparticle also. We have compared with the before and after conjugation, and then the XPSS analysis were confirmed the presence of the zinc oxide nanoparticles. And then when we go to the in vitro cytotoxicity experiment in the control and the zinc oxide nanoparticle, when comparing to the trichoderma beta-glucan, the zinc oxide nanoparticle and beta-glucan conjugated nanoparticle were showed the higher activity of cell cytotoxicity. It's evidenced by the AOEB staining and as well the flow cytometry annexing FITC experiments. So also we tested the antibacterial activity is also similarly found higher with the beta-glucan conjugated zinc oxide nanoparticle, since the beta-glucan and the zinc oxide nanoparticle both, when it's conjugated together, it caused through the synergetic effect against the pathogens, the multi-drug resistant Staphylococcus aureus. So followed by that experiment, we tested in the two kind of experiment. One, as I mentioned, in vitro cell line. Also, we tested in the in vivo antibacterial activity. In the antibacterial activity, we tested in a roundworm, C. elegans. In the C. elegans, we have treated with bacteria as well as the nanoparticle. When the nanoparticle is treated, the bacterial colonization is incubated and healthy worms we have found, and survival were increased, and also the bacterial viability and cells were decreased when we treated with the beta-glucan conjugated zinc oxide nanoparticle. And then another one more study we done, as I mentioned, the aptamer is completely present on the surface. It is a DNA aptamer. It can be used as the immune stimulator also. Some cases it's the DNA transfer, as also it is a ligand to target the cancer cells. So in this aspect, we developed the drug carrier using the chitosan as the nanoparticle by loading the Ernatoc as the cancer drug, being used for the lung cancer therapy. So in this experiment, we find out the characterization by the nanoparticle, TEM, and regular high throughput experimental methods. And also here significantly, I want to explain here that this is the marker. Aptamer conjugation is, as I told, it's a DNA nucleotide. So when we conjugating with this nanoparticle, the aptamer conjugation can be tested in one method is electrophoresis. In the electrophoresis, you can able to see if there is no nanoparticle, so there is no aptamer, so there is no move. Here is the aptamer, so you can able to see there is the nanoparticle conjugated nanoparticle conjugated aptamer can able to see here, because the nanoparticle not move through the electrophoresis. So when the aptamer is successfully conjugated, that not move or just hold on the well itself. So it is confirming the successful conjugation of aptamer. And followed by these results, we can able to see how the aptamer conjugated Ernatoc chitosan nanoparticle is internalized. We have tested in the two different methods. For example, when one is we have conjugated, one we have used for the conjugating some dye, another one is we use the TEM. You can able to see higher internalization were found with the aptamer conjugation through the ligand and the nucleon receptor interactions. Also, the cytotoxicity cell viability experiment were confirmed their higher anti-cancer activity through the optimal conjugation. So also these results we confirmed through the different flow cytometry staining assays. These assays also confirmed their higher nucleus damage and also ROS production and mitochondrial membrane loss through treatment of optimal lactocatosis nanoparticles. So this study we confirmed that the optimer conjugated lactoglucosidase nanoparticle can cause the cancer cell death through inducing the ROS production and triggering the mitochondrial membrane loss by activating the catosine degradation in here. So in this mechanism, the cell death were occurred. This paper already is published. So with this, I concluded that this research may provide us some fundamental information about the trichodermal strains from the ecology to their industrial applications, also the immunonanomedicine. So that it's just a beginning of the experiments or we are going on, and upcoming studies or more advanced experiments are in progress. We will in progress to get more advanced publications. So with this, I introduce here, there was the call for paper. We have editing at presently and collaboration with the Dr. Mubarak Ali. We are editing a special issue. The special issue called Biomolecules from the Fungi for Agricultural and Pharmaceutical Applications. If anyone interested can submit their papers to here. And then also another special issue we have advanced is in marine biotechnology for human welfare and entrepreneurship. This one also been edited on association with the Dr. Mubarak Ali. This also may be more interested to the most of scientists. And with this, I would like to thank my mentor from India, Professor Kadrisan sir, as well as from China, Professor Chen, and in Korea, Professor Wong. In addition, I thank my funding agency, National Research Foundation, as well as NRF and NSFC from China, as well as Ministry of Environmental Forestries from India. In addition, based on our work, we are also seeking the collaboration and internship and online training program, and how to writing the research article, research ethics, and most of the things we are proceeding. And at present moment, we are actively collaborating with several institutions in India. If anyone interested, can be contact through the email or the phone number, which will help us to establish the further collaboration on the research what we are carrying in our lab. Thank you very much for your kind attention. And thank you very much for Dr. Mubarak Ali and organizing committee and all who were responsible to handling this great event. And once again, sorry for the inconvenience. I don't know what is the reason. During my presentation, there was some disturbance. Thank you.
Okay, thank you very much, Dr. Sarvan, for your excellent presentation. That is showing that from the scratch to the advanced study, you have covered all the field almost to establishing the research on the trichoderm especially. If the topic is now open for the discussion, you can raise hands, I can make you to unmute, you can talk, otherwise you can drop your message, question in the chat box. Meanwhile, you can note his mail ID and then WhatsApp number. If you have wanted to establish any collaboration further for it. Maybe it takes some time to type it. Maybe you can wait 1 or 2 minutes. Sarun, you can stop share. Yeah.
Okay.
So any question, clarification from your side, students? Any question in live? Okay. I think they are almost based on your grammatical representation and then diagrammatical, sorry, for diagrammatical representation, and most of the pictures they would have understand a lot rather than asking many questions. So with this, no further question, I'd like to thank Dr. Sharaman for his presentation. This is a certificate of appreciation. Doctor, please, you can accept this virtual certificate from us. And thanks for accepting our invitation and delivering a lecture. Maybe this is even late hours there. You can join and then I have presented. Thank you so much.
Okay, thank you so much for your kind invitation, and thank you all. And as I mentioned, as Dr. Mubarak also mentioned, anytime you can ask any questions related to this research what I presented today through the email or any other way. Thank you so much for today.
Yeah, if any feedback comes, then I can post you in the, maybe personally I can mail you about their because in the feedback it is one of the thing you are requested. So thank you, Doctor. Maybe I can proceed further and end up this today's event. So we are come to the end of this day one. You can find the feedback link here, and also I post in the YouTube live, and also here in the chat box. You can fill it. And you are also invited to join for the day 2 event at 2 p.m. tomorrow, and also for the inauguration of Nano and the Biomedical Association. So successful participation and completion of this feedback link for the both the days will, or the candidate who get their certificate, eligible to get the certificates. So in few minutes, it automatically turn off this window. You can note the feedback link, or you can click that. Okay, thank you so much for joining today, and we are ready to for the initial technical issue. This is unexpected. I hope it will not be happen tomorrow. And have a nice day. Thank you so much.